Hello everyone, welcome back to my video tutorial for multimodal data analysis. Today I'm going to show you how to use the Muon package and perform multimodal data analysis. So if you haven't installed the package, you can use pip install Muon. I installed it already, so let's just load the packages. And we set the opacity as 3, and also I'm loading SC to help me get rid of the warning messages. So first let's change the files directory to the root folder, and we can have a look. Where is my root folder? You can see it's in CITE-seq users. We are going to use the CITE-seq data for today's video tutorial. So I downloaded the sequencing data in the CITE folder in my computer. So you can see it's under CITE-seq users, my username, documents, and CITE. So if you want to practice use this dataset, you can download the data from 10x Genomics website. You need to download the filtered feature BC matrix data, and also I downloaded the raw feature BC matrix data because we are going to use the DBS normalization method for protein sequencing data normalization. So we can set the files directory to the CITE folder. If we have a look at the files in the folder, you can see we have the filtered feature BC matrix and also the raw feature BC matrix. So let's read the filtered feature BC matrix in as M data. And also we read the raw feature data as M data raw. So let's have a look at the M data. You can see the data contain both RNA sequencing data and the antibody tagged oligoarray data. You can see we have 5,247 cells, and in the protein data we have 32 antibodies. And up here you can see for the mu data object, the feature types for the RNA and the feature types for the protein have been added together. So let's have a look at the observation names for the RNA. You can see they are cell barcode, and the variable names for the RNA, they are genes. So the observation names for the protein should be the same as the RNA, they are barcodes. And we can have a look at the variable names for the protein data. You can see we have 32 antibodies. The last 3 antibodies, IgG1 control, IgG2A control, and IgG2B control, they are isotopes control, so we can use the isotopes control for data normalization. As I showed you, the mu data contain both RNA and protein data. We can use M data mode function to analyze RNA data and protein data separately. So let's get the RNA data first. You can see we have the RNA observations and the variables. So now we are analyzing the RNA data. But for all our analysis in the following step, we will save the result in the M data. So first we can calculate the QC matrix for the percentage of mitochondria DNA N genes by counts and total counts. Normally we use the wireline plot to see the N genes by count, total count, and percentage of mitochondria DNA. The MLM package also has a histogram function to see N genes, total count, and percentage mitochondria DNA. Let's run both plot functions, histogram and wireline. You can see we generate the histogram figures for the N genes by count, total count, and percentage counts for mitochondria DNA. Down here are the wireline plot. You can see in the histogram plot we have 3 peaks for the N gene by count, and in the wireline plot we have 3 areas show high density of cell numbers. So each is the same. For the total count we have 2 peaks here, and we have 2 regions in the wireline plot. So normally we keep cells in this region, so we can subset the cells and perform the quality control analysis. So Mu package perform in-place data filtering. This can help you to use less your computer memory. Here we are going to filter the genes that have expression less than 3 cells. The in-place filtering works as the same. The filter genes function in Scampi. So let's filter the genes. Then we perform the in-place filtering for the N genes by counts bigger than 200 and smaller than 5,000. For the total count, it's bigger than 1,500 and smaller than 15,000, and the mitochondria DNA percentage is smaller than 20%. So after the in-place filtering, we can use the histogram and wireline plot to look at the N genes by count, total count, and mitochondria DNA percentage again. You can see now the shapes for the peaks and also the wireline plot are changed. So after the quality control analysis, we can perform the data normalization, logarithmize the data, then select the highly variable genes for the RNA data. Also, we plot the highly variable genes after the data normalization and before the data normalization. You can see the difference, the distribution of the variable genes. Let's see how many variable genes we have from the analysis. You can see it is 1,734 variable genes that we are going to use them for our dimensional reduction analysis and cell clustering. So before we perform the following analysis, we can set the RNA data as raw. Then we can save the original filtered RNA counts. Now we can perform data scaling, PCA analysis. We use different mark genes to show the cell clusters after PCA analysis. This is a PBMC dataset from healthy donors. So we can use CD14 for CD14 positive monocyte, and also if CGR3 shows the CD16 positive monocyte. So we know this cluster contain monocytes. And the CD3D is a mark gene for T cells. NKG7 is a mark gene for NK cells. So T cells and NK cells are in this cluster. we use CD79A as the mark gene for B cells and IRF8 as the mark gene to enable the dendritic cells. So you can see after the PCR analysis, we can identify the cell canisters. But normally we show the cell canisters after non-lineage dimension reduction analysis. So we can have a look at the PCA variance ratio. Then we can select correct PCA for the analysis. You can see this is very similar like the elbow plot in throughout analysis. If we use PC20, will be good enough to perform the neighbor analysis. So let's use PC20 to find the neighbors. Then perform UMAP analysis. Now we are ready to use the latent canister method to identify the cell canisters. I use the resolution 0.1 here. You can see we have 7 cell canisters. So let's plot a figure to see the cell canisters. You can see we have 7 cell canisters. At the moment we don't know what kind of cell types in each cell canister, so we can use the rank genes group to identify mark genes for each cell canister. Here we are going to use the t-test method. Let's run the rank genes groups function. So you can see we identified the mark genes for each cell canister. We saved the mark genes into the unstructured layer as the rank genes group. We have the names, scores, log folder changes, p-values, and adjusted p-values for identified genes for each cell canister. So let's create a data frame to see the mark genes for each cell canister. You can see here, canister 0 has the mark gene LYZ CST3, so they are monocyte. Canister 1 has CD3D, CD3E, IL7R, and CCL5, so they are T cells. And 3 we can see NKG7, so they are NK cells. Canister 4 has mark gene CD79A, so they are B cells. Canister 5 has mark gene FCGR3A, so cells in canister 5 are CD16 positive monocyte, and so in canister 6, cells express the mark gene IRF8, so cells in canister 6 are dendritic cells. So we can make a dictionary for the mark genes identified in above analysis for different cell types in the PBMC dataset. Then we can use the dot plot to show the mark genes for different cell types. Because we want to put the dendrogram on, so we can run the dendrogram analysis first for the latent cell canisters. Now we can run the plot. You can see mark genes for CD4 positive monocyte, mark genes for memory T cells, naive T cells, NK cells, B cells, CD16 positive monocyte, and dendritic cells. So now, according to the mark genes we identified, we can rename the cell canisters as CD14 monocyte for canister 0, memory T cells for canister 1, naive T cells for canister 2, NK cells for canister 3, B cells canister 4, canister 5 is CD16 positive monocyte, and canister 6 is dendritic cells. Following named the cell clusters, we can plot the cells again. You can see now on the left hand side we have the latent cell clusters, and on the right hand side we named the cell clusters. If you have a look here, the color code for each cell cluster is different for the latent cell cluster and the cell types we labeled. So we can reorder the cell types and run the cell plot again. You can see now they have the same color for each cell cluster. So we finish the RNA data analysis. We can have a look at the M data. You can see now in the RNA data we have all the information for the analysis, but for the protein data we just have the variables, gene IDs, and the feature types. And also you probably noticed that the total number of observations in the RNA is different from the protein data now, because we performed the quality control for the RNA dataset and get rid of the low quality cells. So we can use the intersect function in the mu package to select the same cells in the protein dataset as the RNA dataset. If we run the intersect observation function and have a look at the M data again, you can see now we have the same number cells in the RNA data as the protein data. So we performed the cell clustering using the RNA data, so we can use the mu plot function to see protein information on each cell clusters. So we can set the basis argument in the mu plot function as the RNA UMAP. Then we will see the cell clusters by the RNA data analysis. Then we can see, for example, CD3 protein expression and also the gene expression on the RNA cell clusters. Let's run the plotting. You can see we have the CD3 protein information in this cell cluster. They are T cells, and also the CD3G gene information in the T cells. At the moment you can see the gene information is quite strong, but the protein information is quite weak. This is because we didn't perform data normalization for the protein assay. So let's normalize the protein data. There are different methods we can use to normalize the protein data. The easiest one is the CLR normalization method. We used this method in my short video tutorial for the multimodal CITE-seq analysis. If you want to use this method, you can run this code. Today I'm going to show you another method for CITE protein data normalization. The DBS normalization method. It is a method specially developed for CITE-seq data analysis. When we see the variables for the protein assay, we have three IgG antibody as isotopes control. So the isotope antibody controls can be taken into account by the DBS normalization method. So let's get the information for three isotopes antibody control. You can see they are IgG1, IgG2A, and IgG2B. Now we can perform the DSB protein data normalization. So after the data normalization, we can plot a CD3 protein and the gene on the RNA cell cluster again. You can see now we can see strong signal for CD3 protein expression on the T cell canisters. So we canist the cells based on the RNA sequencing data and use the CD3 protein and the gene named CD3G as an example to show their expression for the multimodal analysis. I prefer use the cell canisters from the RNA data analysis because we have all the gene information and it is easier to identify different cell types. But for the multimodal analysis, because we have the protein assay data, you can also canist the cells based on the protein assay. As I showed you for the RNA data analysis, we can analyze the protein data separately. So we can use the data mode function to get to the protein data. We already performed the data normalization for the protein data, so we just need to run PCA, find the neighbors, run UMAP, and also we can use the latent canistering method for the protein assay. Now we can plot the cell canisters according to the protein latent canisters. You can see we have 7 cell canisters. The shape of cell canisters looks different as the RNA cell canisters. Again, we can plot the protein expression on the protein cell canisters. We can use the CD3, CD4, and CD8 antibodies sequencing information to identify the T cell canisters. Let's run. You can see 2 cell canisters express the T cell mark CD3 protein, and the CD4 label the CD4 positive T cells in this cell canister, and the CD8A label the CD8 positive T cell canister. Now we can set the basis as protein UMAP and plot the CD3 protein and the gene again. On the protein cell canisters, you can see CD3 protein is expressed on both cell canisters, and both cell canisters express high level CD3G gene. So as I mentioned above, I prefer to use the cell canisters from the RNA data analysis. So for today's video tutorial, I used the Muon package and showed you how to perform CITE multimodal data analysis in Python. So I'm going to stop for today's demonstration. I hope my video tutorial could help your data analysis. Please subscribe to my channel and share my videos with your friends. Thank you, and I hope to see you in my next video.